Today we're going to be talking about the second step in protein synthesis, which is translation. But before we do that, we are going to review. So tell me, which is single-stranded, DNA, RNA, or both? It is RNA. Remember, RNA is just one single strand, DNA is two strands. Which uses adenine, which is the letter A, as a base, DNA, RNA, or both? It is both. Remember, DNA has A, C, G, and T. RNA has A, C, G, and U. Both of them have the letter A. Which uses ribose as a sugar? DNA, RNA, or both? This would be RNA. Remember, it's called deoxyribose for DNA and ribose for RNA. Here's a picture of those two sugars. They're only slightly different. You don't need to know the difference here. Which is not found in DNA? That would be U for uracil. Remember, RNA has U, DNA has T. Which molecule can leave the nucleus? DNA, RNA, or both? Remember, it is RNA that can leave the nucleus. Which leads me to my next question. How do we get the message from DNA out of the nucleus to the ribosome? We use our messenger RNA. Remember, that's the molecule we make during transcription. It's one strand, and this molecule can travel outside to the ribosome. Do me a favor, create the matching mRNA strand. So remember, mRNA does not have T's, so you should not have T's in your answer. So answer here should be UAA, GUUC. What are amino acids? They are molecules that make up proteins. Remember, each of these little circles is an amino acid. Together, they make up a protein, and that's the goal of the cell, is to make protein. So that's the process we're talking about today. What makes each amino acid structure unique? This is the R group, right? The group on the bottom here. And what are the bonds called that connect amino acids? These are called peptide bonds. Good. All right, we're going to talk about translation. So get out your notebook. Your title can be translation. You'll notice this sounds a lot like transcription, which is what we just talked about. Transcription is where we converted the DNA into the message, the messenger RNA. Now we're going to talk about what happens to that messenger RNA. It's going to go to the ribosome, and what happens there. So how do we get from messenger RNA to protein? Go ahead and write this down. Proteins are made in two steps. The first step is transcription. Transcription is where we're making that messenger RNA. The DNA message gets turned into mRNA. Translation is where we take that messenger RNA, then we're going to bring it to the ribosome, and we're going to turn it into a protein. If you need to continue writing, go ahead, just hit pause. All right, so we're going to talk about the steps. You're going to write down these steps. There are a few steps here that we will be talking about. So how does this all work? Well, our messenger RNA is going to go to the ribosome, and it attaches there. attaches to the ribosome, and the ribosome's going to read the messenger RNA. It's going to read the things called codons, which we haven't talked about yet. I'm going to talk about it on the next slide. Here's my little messenger RNA. This blob is the ribosome. It's actually made of two parts. So it's got this bigger part and this smaller part, and in between feeds the little messenger RNA. Again, pause if you don't, if you still need to continue writing. So I said it reads a codon. Okay, well here's our RNA strand. It's talking about codons here. Can you look at this picture and tell me how many letters make up one codon? It's 3. See how there's 3 letters that make up one codon? So this is what I like to tell students, okay? Think about letters of the alphabet. There are 26 letters of the alphabet. We can rearrange them in different ways and we make words. Codons are like those words, all right? So let's write down what a codon is. A codon is a set of 3 messenger RNA letters or bases. Remember those bases, A, C, G, and U? And these code for an amino acid. Just like if we put 3 letters together, they might code for a word. C-A-T codes for cat, right, in the dictionary. Similar process. It's called a codon because it's a code. The letters code for an amino acid. So maybe we have AAC. That's going to code for a different amino acid than CCG. Remember, our amino acids are going to be connecting together to make that protein. So our next question then, this code. How do I know the code? How do we know what codon codes for each amino acid? So here's an example of a codon, AAC. We want to figure out what amino acid does it code for. Well, here's what we do. We look at the inner circle and we find the first letter in the inner circle. So we've got AAC. So that first letter is going to be an A. Okay, there's our first letter. Once we find that first letter, we block off all the rest of it. Okay, we don't need to worry about the rest of it. We're just going to look in this corner where the big A is. Then we're going to find the second letter, but this time we're going to be looking in the second ring here. So AAC. So my second letter is A. So again, there's my second letter. Now we're going to block off everything else. You'll notice this A is in between these 2 lines. So we're going to block off everything else. And then we're going to find the last letter in the third ring. So C. So AAC. So this would code for asparagine. That's the name of an amino acid. You don't need to know what it looks like. Just know it's a molecule. It's an amino acid. It makes up proteins. Okay? You try. So I want you to find UAG. Remember, find the first letter in this inner circle. Okay? Then find the second letter and then the third letter. So you should have gotten stop. Well, that sounds silly. How did we get that? Well, U is right here. Okay, so we only look in this corner. Then we've got A and G. Stop means to stop making the protein. Okay, so that's how the cell knows. Okay. All right. And one more. What does CCG code for? So find our first letter, C, second letter, C, third letter, G. Proline. Good. All right, so you didn't have anything to write down for that, but now we're going to finish with our steps. So step number 2. Once we know what it codes for, remember our ribosomes reading the letters? We're going to have a helper molecule. It's another type of RNA. It's called tRNA. It's going to actually bring the amino acid. So even though we know what the amino acid codes for, we have to bring the amino acid there. So So our tRNA is going to bring the amino acid to the ribosome. tRNA stands for transfer RNA. You can kind of remember it because it kind of looks like the letter T, kind of, right? So tRNA, you'll notice it's got these amino acids on the top. Remember, if we connect them together, they make a protein. That's our goal here in the ribosome. So how does the tRNA know where to go then? That's our next question. Well, the tRNA knows where to go because it's got letters that connect. Okay? These letters, write this down. These letters are called an anticodon. Anti means not or against. So basically, it's got 3 letters that match with the correct codon. So if we have AAA, okay, on our messenger molecule, it would match with UUU. If we've got GGG, it's going to match with CCC. So tRNA is RNA. So what letters would we use for tRNA? We would use A, U, C, and G, because remember there are no T's in RNA. All right, so I want you to take a second and I want you to choose the answer that has the correctly matching tRNA letters. So you'll see right here, we've got in our mRNA AGU, CGU. I need you to tell me what letters would match there. So you should have gotten UCA, GCA, okay? All right, we are almost done with our steps. So we've got step 3 and 4 and then we're done. So step 3, the amino acids are connected together by those peptide bonds. Okay, those are the little links in between those amino acids that we see. You'll notice as the tRNA is dropping off those amino acids, it forms those bonds. And last but not least, okay, the process continues until we reach a stop codon. Remember on our little chart, there was a set of 3 letters that codes for stop and tells the ribosome to stop making the protein. All right, we're going to do one example and then we are all done. So you try in this example. Choose the correct answer for the mRNA. So A goes with U, because remember in DNA it goes with T, but there are no T's, so we got to put a U. C with G, C with G, T with A with U, G with C. Okay, now that we have the messenger RNA, I need you to tell me what are the correct amino acids that go with this mRNA. Now, if you're confused, remember, we always use the messenger RNA for our little chart here. You'll notice our chart doesn't have any T's, so we can't use DNA. We've got to use these letters. So a set of 3 here, first set of 3 is one codon, second set of 3 is another codon. So if I do U, find my second letter G, and then G, tryptophan, okay? And then our second one ended up being isoleucine. Now scientists are lazy, so they don't usually write the whole word out. You can just write the first 3 letters, okay? So on worksheets that we're gonna be working on, you can just write the first 3 letters. All right, that is it. Have a fabulous rest of your day.